So this is our student session of the MGI Symposium, and we have three excellent speakers lined up. So the first speaker is Professor Santhi Ganesh from Internal Medicine and Human Genetics. She is going to tell us about leveraging the MGI resource to define polygenic increases on sex-biased cardiovascular diseases.
Sex-biased cardiovascular diseases and a couple of diseases that I spend a lot of my time speaking about. We use NGI quite a bit. Our cohorts are now targeting NGI, so we're excited about that. As far as disclosures, we have extramural funding and work with patient advocacy groups that's on the SOCDIT, and we have filed invention disclosures on some genetic risk assessment tools, some of which I'll talk about. So what I thought to talk about in the 15 to 20 minutes today is sex-biased cardiovascular disease in the traits, muscular dysplasia, spontaneous coronary artery dissection, which maybe you haven't heard of either of those two before. I'll explain what they are, and blood pressure, then what our current genetic understanding is and how we're using this information. So just a general thought about sex-biased cardiovascular diseases, I would say most, if not all, cardiovascular diseases have various sex biases within them. If not their prevalence, then their presentation, the long-term trajectories, outcomes, and so forth. There's varies in all of these categories that I've listed here where the same risk factor may lead to a different outcome according to sex, biological sex here. Clinical disparities in terms of our recognition of disease, which is something that we have seen for the diseases that I'll talk about today. But also how interventions are performed, what interventions are considered at what time point, what the outcomes are. Many female-biased diseases have not been studied in the same depth. This is an argument is made to funding agencies like NIH have been quite successful at pointing out in some areas of real need. And just to keep in mind that there are sex biases in terms of all of these things, but there are also sex-specific diseases such as those occurring in pregnancy. So I'm going to talk about fibromuscular dysplasia and I mentioned spontaneous coronary artery dissection. And this is due to a process in the blood vessel wall. So probably most people here have heard about cholesterol plaques, atherosclerosis that can develop in the vessel. This is a completely different cause that is not cholesterol driven, but instead is how the cells are organizing themselves, how they're functioning, how they're making the extracellular matrix. And the kind of macroscopic view of what happens to an artery affected by this process, if the normal is over on the left, and you have this arterial bleeding is what it's called, because when you inject dye in the middle, you get this recurrent stenosis and area of dilation. So it looks like a string of beads or a string of pearls. That's how it's sometimes referred to. You could have aneurysms in 20 to 25% of patients and arterial dissections. It could either a tear in the vessel wall or spontaneous separation of the layers. The end result is that vessel isn't able to deliver the blood flow it's supposed to, and there is lack of blood perfusion to the organ. So in the case of coronary artery dissection, it's the heart, and it's a cause of myocardial infarction. And so the manifestations can be wherever these vessels are affected. If it's the kidney, it's resistant hypertension because the kidney does not like to get too little blood flow, and we'll do things to correct that and so forth. So this is a picture of what it actually looks like in a patient, an angiogram with the two renal arteries coming off. And you can see that it's lumpy and bumpy, and really it should be very smooth. This is my only gross pathology picture, but this is what it looks like. You know that there's this corrugated appearance to the vessel. That is not normal. It should be a smooth surface. And when you look histologically, there's a lot of collagen and glue on this trichrome stain and thinning and thickening of the arterial media, which is a middle muscular layer of the branch vessels of the aorta, which are the vessels that tend to get affected by this. 90% of patients are women. The true prevalence is not really known. You'd have to do CT studies from people's heads through their pelvis and still miss things like the arms and the legs. We don't have that in any population basis. So we have donor angiograms when people donate a kidney, and based on that, the prevalence is estimated to be 3.3%. So this disease used to be thought of as rare, kind of very unusual cause of severe hypertension in a 20-year-old woman who was presenting out of the blue with severe hypertension. That was kind of the classic story. And now we're saying it's a pretty prevalent cause of heart attack, stroke, many cardiovascular manifestations, and definitely above any of the definitions of rare that like the NORD organizations and so forth would use. The estimated number of cases probably at least per SCAD closer to 800,000 a year that we're aware of. So we have here at Michigan been able to do some really special things with our patient cohorts. And this is kind of the paradigm for our research program where we're enrolling patients, and the Taubman Institute has very generously supported our efforts in this area to get really good phenotyping. This includes some of that angiographic imaging. We think about what are the kind of important clinical questions in those cohorts, collect patient samples. We're doing GWAS and exome sequencing, targeted sequencing, thinking about what molecular phenotypes may make sense, and then carry forward some of these discoveries into functional studies. So this has been really fruitful, and recently have been able to secure good funding for different components of this triangle. So we're pleased to have the progress we have now. We've been enrolling patients here at Michigan for about 10 years in total, and have done our first GWAS of FMD, which is, the numbers are shown here. The University of Michigan group is combined the cohort from Cleveland Clinic, and we accessed MGI for controls, which I'll describe. We also had to do a meta-analysis combining a few hundred cases from multiple different places, and eventually did have the first GWAS meta-analysis for FMD that identified these 4 loci. And, that's very exciting. And we see things that are potentially very interesting. You'll see some recurrent themes, and I won't probably have time to get into too much of the specifics, but there's a lot of biology within the genes that have been suggested. Co-localization analyses strongly prioritize LRP1 and factor 1 in particular. And there's some really interesting aspects about these associations that I'll try to draw out. So that's when we think about, on this allele frequency versus effect size spectrum, down here in the right lower corner for common diseases, where we're getting at things like genome-wide association study. We also have had some success looking across the spectrum here. I think my FMD slide was actually a little bit early, so let me show you this GWAS workflow that we have used, where we've enrolled our cases from multiple sites, and then we're using MGI. In our initial phases, we actually implemented an algorithm that excluded vascular disease in healthy control individuals, and we were able to get to several thousand control and have highly imbalanced case control sample sizes, and used SAGE for our association analyses. And just to show, we were able to submit samples to the genomics core and get genotype from doing the sample QC infusion together, and conducted GWAS and PRS analyses. For our GWAS at SCAD, we actually have worked with a Canadian registry that has 22 sites across Canada and a few sites in the United States. University of Michigan is one of them. And in our first GWAS, these are very small numbers for a GWAS, but this is what we had. We were able to conduct a discovery analysis, a replication, meta-analysis, and then do some follow-up work in our FMD cohorts and KitBioMag and the Patriot program. And this is what our first GWAS looked like with just 270 cases. And it's really because this chromosome 1 hit has an odds ratio of close to 2. We replicated that in our samples and now replicated in other samples, and now completed a meta-analysis with other groups and have more findings. But this first GWAS was really very instructive. This first top hit for chromosome 1, this was the co-localization analysis that very strongly prioritized this gene Adams-like 4, which is about 300 kb from the index SNP. Posterior probability was 98% or so, pretty strongly suggested, and it turns out this is a really excellent candidate gene for the extracellular matrix. It's involved in regulating the function of some of the enzymes that put collagens together and so forth to make some of the bundles of collagen and fibrillin in the exosome wall. So we have Minli Yang is the graduate student who completed this GWAS, and we've now taken these findings into the lab and gives the direction of effect that it's the SCAD risk allele lowers the expression of Adams-like 4. So we've done knockdown experiments and some bioengineering types of experiments where we can grow cells into 3D and stress them into failure and look at their tensile properties. And there's actually quite a lot going on with the coronary arteries and muscle cells. So that's very interesting. Cameron Kenneth is the fellow who is doing those studies, and he'll be at the ASH thing next month, or fill out 5 or 6 days. So from that SCAD GWAS, we did construct a polygenic score from the top 7 SNPs and used that PGS to go into the UK Biobank and conducted a CWAS. A couple of our very interesting findings is a very strong positive association with migraine headache and a inverse association with atherosclerotic form of MI. So atherosclerotic MI is very prevalent, but more prevalent among male individuals compared to female. Migraine might be more female biased, but we saw consistent effects in both male and female individuals. And some of this was not a surprise because many of our scan patients have migraine headaches. And what we've seen so far, and I think it's mostly anecdotal for our FMD and SCAD cohorts, where some of the loci we had overlap, the factor 1 locus, the LRP1 locus, those overlap. What we are seeing is that this inverse association with athero, we see it in the families. Many of the family members have a lot of longevity. We see patients who've had a coronary dissection or heart attack in their 50s, or a history like that they're in their 70s and 80s and up to 90, doing actually quite well. So that's quite intriguing. We looked at this from the standpoint of what can we learn about familial relative risk, but I thought there was something instructive here and interesting to talk about. Alex Katz is a former postdoc from the lab, where his study was to look at our family collections of FMD, and he reviewed 73 probabs and all of the first-degree family members. In total, there was 536 family members for those 73 individuals. And our question was, what's the relative risk for FMD? At that point, we didn't know if there are common genetic factors or the alleles or what have you. We're tough just if they're diagnosed phenotype and continue this angiographic imaging. And in the daughters, it turned out that many of them had workups and their relative risk is 3. And so that was notable. In doing this research, Alex said, is it interesting to use that some of the fathers have abdominal aortic aneurysms? We thought, well, that's not really FMD, it's something different, but maybe count up the numbers. And so when he did, he saw that the fathers had a relative risk of 2.3. And so we thought that's interesting, but really it's not that many affected individuals who are driving this signal. So he went to ASHG and reported this data, but we didn't publish it for a while until we could use some of the polygenic scores that were under development. And this idea of in female individuals, maybe we have one set of diseases that then with the shared genetics have a completely different manifestation was the idea. So we went to the Million Veterans Program where they have a large collection of genotyped and phenotyped individuals with a strongly male biased cohort because of the makeup of the PA medical system. And when a PRS that they had developed for AAA was tested for FMD, the odds ratio was there, 1.5 is modest, but a positive association, and may converse, the PRS for FMD was associated with AAA. And so after that, we said, well, there does seem to be a link here, and actually there is a shared association of that LRP1 locus for both diseases. So I think this raises some really interesting biologic questions of why there might be these tissue-specific effects and differences in how the tissues are responding. In FMD, we think of it as the vessel growing inward, and the aneurysm as the vessel growing outwards. And so we now have some work ongoing to try to understand this in terms of the biologic drivers and our hormone-sensitive elements that are driving gene expression. And are there links to other related traits? So, for example, hypertension is a risk factor for some of the complications of FMD. Hypertension is definitely a risk factor for abdominal aortic aneurysm, any aneurysm. And so I think this morning Cheng Xu presented some of the work on our sex differences of blood pressure traits, and he's gone into the UK Biobank and done completely sex stratified studies of blood pressure. Here I'm showing systolic blood pressure, but we also did this for diastolic blood pressure and pulse pressure. And you can see there are some hits that in purple for the female group in blue, for the male group it's black, for the overall group, that there are some shared signals. There are more signals you find with the higher sample size and better power, but there are also some distinct signals. And so we actually did a lot of analyses. Min Lee did this work to look at the relationship of the blood pressure determinants with cardiovascular disease as a trait. And I thought I'd pull out the FND and SCAD for this talk, where what we see is the PGOs of the pulse pressure is what's shown below. And this is the x-axis is cut off here. I apologize for that. But the z-score is was plotted on the x-axis, and blue is for male, purple is for female, and the orange is the combined sex pulse pressure GWAS-derived polygenic score. And I just show this to say that there are actually some dimorphic effects in terms of the SCAD associations. We have done other work that shows there's dimorphic effects on aneurysm of the aorta. For systolic blood pressure, there are a whole series of differences. So as we think about blood pressure is a major driver of many cardiovascular diseases. It's a risk factor for most of the big ones that we think about: stroke, heart attack, heart failure, arrhythmia, atrial fibrillation, and so forth. There's the idea that there are sex differences is quite intriguing. Other work that isn't shown on the slide is that when we look to see what transcription factors are enhanced to the low side that come out, we have estrogen signals and so forth. So it seems, that there's probably some very important hormonal biology to think about. Min Lee will be presenting this work in more detail. So I have 1 minute left, so I'm going to choose to focus on a quick story, which is that we did, through sequencing of our patients, get to find one collagen 5A1 recurrent variant. This is our 4 probands, assembled through clinic, my clinic and a clinic in Ontario, and then reaching out to other clinicians, and found this variant that was not seen in NOMAD. What we were able to do, the COL5A1 gene is for classical EDS. This is just the LD in the region containing that very rare variant, plotted against 1,000 genomes, the genome-wide SNPs, the ancestry of those 4 probands, suggested to be Central European. And then we could phase haplotypes out of that data, which we did in 1,000 genomes data as well as MGI. And we're able to show that there's a shared haplotype with this variant, suggesting that it arose on a founder haplotype and then was passed down. The mean age of onset and diagnosis was age 50. So you can imagine how that was passed along, and the variant was upgraded to likely pathogenic, and this is the first clinically actionable finding. There's kind of a cool story that I'll just say very quickly. We thought, could this be a modifier for the occurrence of SCAD in FMD? We know that the polygenic score for SCAD associated with the occurrence of SCAD among those of FMD, which is shown in this top red bar. When we looked at the presence of COL5A1 variants that didn't quite rise to the level of significance as the first variant we found, we did see that the presence of potentially damaging variants, CAT score greater than 20, no matter low frequency less than 1%, were associated with the risk of dissection. So this is highlighted to us through the GWAS in this work, the role of the extracellular matrix in the blood vessel wall as a driver for these diseases. The idea that some of this could be under hormonal regulation, that there's differences in blood pressure regulation that is driving the risk for your blood vessels splitting apart, basically, is something that we're exploring now. So I'll stop there. This is the group. We have many people to thank. We're very fortunate to have a great collaborative group, a whole patient study arm, and great clinical collaborators. They're shown on this slide. Since there is the sex difference, and we know, at least for some of these, there is estrogen signaling thing enrolled. You analyzed, have you split the female group into pre and post menopause, and do you get more information in regards to genetic risk once you have that? Because we do know that there's a lot of protection in pre-menopause, and so you may be missing something if you're grouping them. Yeah. Maybe you're the fair one. Oh, that's bad. So we did that. And I mean, there are these important hormonal effects, we think. The problem is when we split our sample into those 2 subgroups, I think we lost power, essentially. The protection that you're describing has been it's for atherosclerotic disease, we think that what's going on here is more times of hormonal instability or flux. So pregnancy and perimenopause, that's when the patients are presenting with these dissections. So, same issue with hormones, that maybe how we think about it needs to be accounting for that.